Hello nerds, welcome to Ahmad Coaching and I'm your teacher Dr. Anam. Today we are going to study about carbohydrates digestion and absorption. The digestion of carbohydrates starts from the mouth. In the mouth we know that saliva is produced and saliva contains several types of enzymes. One of the enzyme is thylline, which is also known as salivary amylase. So what the salivary amylase or thylline is going to do is going to convert the starch into maltose. We know that starch is actually made up of glucose molecules which are joined together. So this is just going to break down the bond and then we will have the maltose. The maltose is actually made up of two glucose molecules joined together. This enzyme is going to work at the pH 7, which is in the mouth. However, only a small amount of starch it will be digested in the mouth, and rest of the starch it will go from the mouth to the stomach. In the stomach, no digestion of carbohydrates will take place because in the stomach no enzymes are present which can digest the carbohydrates. So the food it will move from the stomach and it will enter into the small intestine. The small intestine is divided into the three parts: duodenum, jejunum and ileum. In the duodenum, when the food it will enter, it is further digested, but there is no enzyme in the duodenum that can digest it. The enzyme which is digesting the starch is coming actually from the pancreas. So pancreas is going to send its secretions into the duodenum, and the starch it will be digested into maltose by the help of pancreatic amylase. Again we have this glucose and glucose bonded molecule. What will happen then? The food it will move from the duodenum and then it will enter into the second part of the small intestine, which is known as jejunum. In the jejunum we know that the wall of the small intestine is actually not a straight one, but it contains several folds. The folds are there so that the surface area it can be increased and the food it can be absorbed easily. The further digestion of the molecules will take place in the jejunum. Here we have this brush border. What is actually the brush border? That in the brush border we have these small cells here, and these small cells they are known as microvilli. So the whole fold is known as like this. The whole fold is actually villi, and with the villi we have the small like these cells, and they are known as microvilli. So this is villi. On the microvilli we have several enzymes attached to it. This whole system is known as brush border. Why brush? Because the shape is like a brush, and it is a border to which the enzymes are attached. What will happen? The food which has entered into the jejunum is going to be digested by the enzymes attached to the brush border. So there are so many microvilli with the lining. The food is going to be digested. Why? Because the enzymes, we know that food is now substrate. The enzymes they are going to convert it into the products. So we have the maltose, and this maltose is going to be digested by the maltase enzyme, and we will have the glucose molecule. Like we know that maltose is consisted of two molecules of glucose, so it is broken like this and we have glucose molecules. Similarly, the lactose sugar which is present in the milk is going to be digested by the lactase enzyme, and we will have one glucose and one galactose. Similarly, the sucrose, which is actually a sugar we consume, or the table sugar we call, it is going to be digested by the sucrase enzyme, and then again we will have glucose and fructose. So in the jejunum we have glucose molecules, we have galactose molecules, and we have fructose molecules, and all these enzymes they are attached here. So what is actually happening, that the substrate is going to be attached here, and the enzyme is going to break it, and the product is going to be removed, and the product is actually glucose, galactose and fructose. These small molecules, now they are ready to be absorbed. At this point the digestion of carbohydrate is completed. So the next step is the absorption of these molecules. The maximum absorption takes place in the ileum part of the small intestine. In the ileum, we again have these microvilli which are attached with the villi. What will happen, that there are special transporters. The transporters, they are actually the proteins which are present on the microvilli. So we have fructose, we have galactose, and then we have glucose. All these molecules are now in the ileum. The fructose is going to be transferred from this lumen into this cell, which is known as enterotypes, through this GLUT5 transporter. So what will happen, that this fructose is going to move from here, and then it will enter into the cell like this. Then the glucose and the galactose, these two molecules, they are going to be transported through SGLT transporter. Now the mechanism behind this transport or this absorption is that inside the cell the sodium concentration, it starts to drop, while in the lumen the sodium concentration is very high. Why it is very high? Because when we consume food we are taking salt, and salt is sodium chloride. That is why the lumen has high concentration of sodium, while the sodium inside this cell is transported out of the cells continuously. So because of the decreased level of sodium inside the cell, the sodium from the outside, it will start to move into the cell, and as it will start moving into the cell, it is going to take the galactose as well as the glucose with it. So as the sodium will move, the galactose and glucose, they both will move, and inside we have galactose and then we have glucose. So this is how these small molecules, they are going to enter into this cell. This SGLT is actually sodium glucose transporter. So it is going to transport sodium as well as glucose and galactose. Now when all these small molecules, they are inside the cell, they are going to be transported out of this through the villi into the blood vessels by the help of the GLUT2 cells. Here we can see this is the transporter. The transporters, they are actually the protein. So this is actually protein which is known as GLUT2. This is going to transport fructose, galactose as well as glucose out of these enterocyte, and it is going to enter into the blood vessels. It is going to enter into the blood which is passing through the villi, and these blood vessels, they are going to combine together to form hepatic portal vein. So in this way the glucose, galactose and fructose, they are going to enter into the liver. So here we have fructose, glucose and galactose. In the liver, these molecules, they are going to be utilized according to the requirement of the body. Maybe they are going to be converted into the energy by the process of glycolysis, or gluconeogenesis is going to take place, or maybe the glucose is going to be stored in the form of glycogen. So in this way, the food which contains the carbohydrate is going to pass through all these parts of the digestive system, and finally it will reach into the liver in the form of fructose, glucose and galactose. So that was all about carbohydrates, digestion and absorption. If you have any further question related to this topic, you can ask. If this video was helpful to you, please like it and share it with your friends. Also subscribe to my channel. I will see you in the next video.